Good afternoon. Thank you for joining us for this afternoon's session that's dedicated to oncolytic viruses. This is a cancer application. My name is Nino Chioca. I'm the current chair of neurosurgery at the Brigham and Women's Hospital, and I've been working in the area of oncolytic viruses both preclinically and clinically since the 1990s. This session is called Oncolytic Viruses, Treating Melanoma and Beyond. And we have a formidable group of panelists. We have Dr. Robert Coffin, who is the current CEO of Replimune, but was a previous CEO of BioVex that brought TVEC from invention all the way to clinical trials and its final commercialization is Imlygic. Then we have Dr. Roger Perlmutter, who is the former CEO of Amgen when they acquired BioVex and Imlygic, and then Merck, and currently is the CEO of ICON. We have Dr. David Reese, who's the current Executive Vice President of Research and Development at Amgen, and then Dr. Ann Silk, who's a physician and assistant professor of medicine at the Brigham and Women's Hospital, who actually takes care of patients with oncolytic viruses and uses it in our practice. So thank you for being here. We will start off right from the start, and I will ask a few questions. And I was going to start off with Dr. Robert Coffin and Dr. Roger Perlmutter, who were the two of the key figures that brought TVEC from concept all the way out to commercialization. And so I will ask both Rob and Roger to maybe paint in broad stroke the history of TVEC and how it became Imlygic and how it got to commercialization. Rob.
Yeah, probably most sensible I start because the history really started back in around 2002 at a company called BioVex, which was started up in the UK. And around that time, we had seen that there was promise for oncolytic therapy, particularly as a means of directly killing tumors, essentially a sort of cytotoxic therapy. And at that time, we thought that we could combine the ability to directly kill tumors with an immune therapy approach to try and provide vaccination and systemic benefit to patients, as well as just local control. So what we were trying to do back in 2002 was essentially combine the best aspects of cancer vaccines, which had looked promising, particularly a GVAX type vaccine, which expresses GM-CSF, with an oncolytic virus. And that was really the theory behind TVEC, combining a potent oncolytic virus, which we based on herpes simplex virus, with the expression of GM-CSF to hopefully induce a systemic immune response, pardon me, against the patient's cancer as a tumor is killed by the virus to provide overall systemic benefit. So at BioVex, we took that concept through phase 1 studies through phase 2 and had promising evidence of activity, particularly in melanoma and head and neck cancer. And we were midway through a phase 3 study in advanced melanoma when we began our discussions with Amgen, who ultimately purchased the company in 2011. And together with the the BioVex team, ultimately achieved FDA approval of TVEC in 2015.
Roger,
and picking up from what Rob said, I think it's important to recognize that Rob himself, his contribution to this was enormous, and he had been very active as a virologist in the UK for a long time. I was familiar with his work and then became familiar with what they were doing at BioVex. When I saw the data that they had obtained in phase 1 and phase 2 studies by direct administration of TVEC intratumorally, recognizing, yes, this is a challenging and difficult way to have to administer a therapeutic. On the other hand, it's worth remembering that at that time, we really didn't have alternatives to chemotherapy for treating aggressive malignant melanoma. Relapsed malignant melanoma at the time had terrible survival. And so this seemed like an opportunity worth exploring, particularly because in the phase 2 studies, there was data that demonstrated that not only the injected tumor samples, but some of the other tumor sites would shrink. And the response in these other sites, the abscopal response, as it's referred to, I found particularly impressive. So it made sense to me that Amgen, which of course was a quite large company with a lot of capability, could take on this project and work together, taking advantage of all the work that had been done by Rob and his colleagues at BioVex, and bring the vaccine, in essence, over the finish line. I should say that at the time within Amgen, there was not enormous enthusiasm for this because of the heterodox nature of the treatment. And it remains the case that this is not necessarily so easy to administer and hence to commercialize. Nevertheless, the results speak for themselves. There's no question that using this approach with a cytolytic virus that has been modified in such a way that it prefers to replicate within the tumor, and taking advantage of what we now understand about immune responsiveness to tumors, a meaningful therapeutic opportunity could be created.
Roger, you discussed the difficulties with commercialization and manufacturing. Maybe the next question is for David, who has been involved at Amgen with Imlygic and trying to get this to patients from 2011 to 2015 and beyond. Can you discuss that a little bit more?
Sure. As Roger and Rob have mentioned, this requires intralesional injection. It seems formidable at first. What we have found is that, there's an education period that clinics can then become quite adept at administering the agent. In many cases, nurse practitioners will administer the agent under supervision as the clinic becomes familiar and makes it routinized. But it does highlight one of the ongoing challenges in the field, which is, can we get beyond the requirement for, at some point, intralesional injection? Will systemic administration of oncolytic viruses, for example, become a reality at some point? Can we overcome the various technical challenges that poses? I'm sure that will come up in the remainder of our discussion. So this is not a therapy for every center. The ones that have adopted it, in our experience, are quite enthusiastic, and they have seen patient benefit, and it's become a routine part of the armamentarium as a monotherapy treatment.
We talk about patient benefit. The next question is for Anne. Anne, you're a physician that actually treats patients, but you're also involved in clinical trials. How do you go about deciding between which patient you give the oncolytic virus versus other treatments?
So currently, I use immune checkpoint inhibitors as first-line therapy for all of my melanoma patients and my non-melanoma patients. But there's certainly the right type of patient that makes sense for whom to use T-VEC first. And as an example, some patients have significant autoimmune disease and they're on immunosuppression, and they have a risk of exacerbating their autoimmune disease by taking the immune checkpoint inhibitor. And for a patient like that, T-VEC is a viable alternative for first-line therapy. I would add that it would make the most sense for a patient who has mostly injectable disease with little or no visceral disease, because although T-VEC does have a systemic effect, it's not as strong as its direct effect.
Thank you. So let's inject a little bit of controversy. It's been 2015 was when T-VEC was, or Imlygic was approved, and for melanoma. When are we going to see an expansion to other cancers? Where are we going to go with oncolytic virus? And when are other cancers going to show evidence that oncolytic virus will be efficacious? Maybe we can start off with Rob.
Well, I certainly don't see the use of oncolytic immunotherapy as being limited to melanoma or skin cancers in the future. I think the modality, which is inducing a systemic immune response against the patient's own cancer, which is not necessarily there before in any great amount or necessarily at all, is clearly necessary for many of the other immune-based approaches to treating cancer to work. They actually require a pre-existing immune response to the patient's cancer to have any effect. And I do see oncolytic immunotherapy as being somewhat unique in the field, as having the potential to induce a potent systemic immune response, which one then may combine with other forms of immunotherapy to potentiate that immune response and do better than either agent would do alone. And certainly at the company we now are working at, Replimune, our ambitions are very much beyond skin cancer. But we do recognize that we need more potent viruses to both kill tumor more effectively and also induce a more potent systemic immune response, which we're doing in various ways. But having done that, our hope and expectation is that we will find utility for oncolytic immunotherapy across a broad spectrum of oncology, including some of the more larger and more difficult to treat tumor types, including things like colorectal cancer, lung cancer, breast cancer, et cetera, once our therapies have been even further established in skin cancer.
Roger, what do you think it will take to get oncolytic viruses as first line or even second line treatment for other cancers?
Well, what's really changed, of course, since the initial studies of TVEC is that we've become aware of just how powerful systemic immunotherapy is. So PD-1 directed therapy, which of course is first line for a large number of tumors, for treatment of a large number of tumors, is dramatically effective. But when I say dramatically effective, it's important to recognize, just as Rob says, that response rates in those tumor settings really need to be improved. I mean, typically on the order of a quarter to a third of patients will respond to systemic therapy with an antibody directed against PD-1 or PD-L1, and with different indications for the different agents. For Keytruda, which is the PD-1 directed antibody that I had the privilege of helping to develop, an extremely broadly active molecule, but just as Rob says, what it does is reveal the pre-existing immunity that's present in a substantial fraction of cancer patients and permits then the immune system to destroy the tumor. It does not stimulate primary immunity in a patient who lacks that. And the hope is that oncolytic viruses would turn into something that could be used in combination with systemic immunotherapy. It is important to mention, we haven't done that yet, but we should, and that is that oncolytic therapy is extremely well tolerated. So the safety profile for oncolytic viruses, as revealed by TVEC, is fantastic. We just need to couple that with something that amplifies the efficacy signal, and I think that is to come.
Yeah, I think that's a really important point that you raise, Roger, that the safety profiles have been quite good. That permits one to envision a lot of potential combinations. And as you and Rob were saying, there are really two core challenges in a sense. One is to take immune susceptible tumors and see if we can enhance the response to agents such as checkpoint inhibitors. The other is to take so-called cold tumors or generally immune resistant tumors and see if we can actually render them immune susceptible to things like checkpoint blockade through the use of an oncolytic virus. And so I would see those as kind of two broad avenues to guide our thinking as we move forward.
I mean, you bring up the point of safety, and I think it's amazing that when oncolytic viruses first came on board, there was real concern about safety, about viruses going awry and causing all sorts of pathogenic effects. But our experience has been that they've been extremely safe, more so than other therapies and drug therapies or antibodies in a lot of senses. Dr. Silk, is that your experience as well? Can you talk a little bit about safety and how patients tolerate injections of these viruses?
Yes, definitely. I think that patients have flu-like symptoms, which last 24 to 48 hours, and then they can go about their daily activities. It's a very, very uncommon that a patient who is being treated with TVEC would need to be hospitalized, but it happens often that patients on immune checkpoint inhibitors do need to be hospitalized for serious systemic immune-related adverse events. There is a fair amount of counseling that goes into making sure patients understand safety precautions, keeping the tumor covered so as not to infect household contacts. But historically, that's not been a problem, and it just requires a little bit of time, which can be done by physicians, mid-levels, nurses, to explain that you treat it like any infected wound. You want to keep it covered. You don't want to touch it. So the safety profile is really good. And again, it's particularly important for patients with pre-existing autoimmune diseases, because for them, flaring their autoimmune disease is a real concern.
Rob, I was wondering if you could speak to all the budding entrepreneurs or academic individuals that want to start NewCos in this area, in this field. You've started one and you were very successful. You have another one now. What does it take? And what does it take particularly to have people like Roger listen to you and basically ultimately help you out with the commercialization of your virus or your product?
I mean, to address the sort of last point first, what really is going to gain the interest of a larger company to work with you is the data. So one needs to focus on generating data and always follow the data and remain true to the data throughout the period of developing your product candidates. But really at the beginning, one has to be prepared for a long road. These things take a very long time. Clinical development is by its very nature takes a long time, and it may well take quite a considerable period, well past phase one, until one has convinced others, including investors and other companies, that one really has something which is potentially useful. You yourself may be convinced from the outset that yes, you have something useful, but it's very likely to take a lot longer to convince other people who may perfectly reasonably be more skeptical. So be prepared for a long road. And also, in a related point, one really does need to have dogged determination and not give up and not stray from the critical path. In reality, it's very easy to be diverted down side roads or places which aren't true to the critical path because they're potentially scientifically interesting. But it's always very important to stick to the critical path, what the real end game is, and not get diverted in such ways. So dogged determination, be prepared for a long haul, and always follow the data. Also know when to give up and not move forward if the data doesn't justify it.
Roger, what attracts you to invest or acquire a company like BioVex or any future company in this area, in this field? What are you looking for as you try to make that decision?
Well, it is, in fact, the reciprocal of what Rob was talking about. A company that has demonstrated a long-term commitment to an idea and that is, not evangelical about it, but at the same time, in a very circumspect way, in a very conscientious way, has explored the underlying science and developed a justification for pursuing it. That's really what you want. There are many things, of course. I mean, for David and now at Amgen, for myself, in a variety of investor contexts, lots of business plans come before us and lots of preliminary results. What we don't like to see is people who stretch the truth. We know how difficult this is and how hard it is to prosecute a phase 3 program and then to build from that into a drug that can bring real benefit to the patients whom we all serve. It takes a very, very long time, and we like small companies that appreciate that.
Thank you.
Yeah, I would really echo that. We ask a simple question: what's the therapeutic hypothesis? What is the evidence, the quality of science that you've brought that supports your ability to prosecute that therapeutic hypothesis? What are the unknowns? What are you doing to address the unknowns and when? And what are the risks? And someone who can explain that in a very straightforward fashion, I think, gains a lot of credibility.
Can I just add a couple of further points? I think one also in an earlier company does need to be thinking about the commercial challenges and realities early on, particularly with the new modality, which has not had an equivalent before. One is breaking down barriers, and that's not only scientific and clinical barriers, it's also commercial barriers to success, some of which David touched on. With T-VEC, it is just injected, which is a straightforward thing in reality, one would imagine for any physician to do or nurse. For deeper tumours, we use imaging guidance, which is exactly the same as taking a biopsy, but in reverse, and therefore any radiologist can do it. But really, it's an education issue and making the product as easy as possible to use once ultimately approved. Another aspect which one certainly should not forget about, and which we probably did better at Biovex than the commercial point, is manufacturing. Manufacturing is critically important, and in our view, it was extraordinarily important to have got that entirely under our own control, not as a contract manufacturer, early on, such that we had our own commercial scale manufacturing plant ready to go for phase 3 and for commercialization. And that's the same at Replimune, where we also have our own manufacturing facility, which is obviously learned from the experience of T-VEC2, which is a critical part of the overall picture.
David, you talked also about the challenges of manufacturing, but also stability and of the T-VEC or Imlygic product. Can you expand on that a bit?
Well, certainly, manufacturability and all of the, supply chain considerations, both pre and post getting the product into the hands of treating physicians and to patients are things that we think about. I think one thing that the pandemic has illustrated to us is that, our colleagues in manufacturing are often the unsung heroes, and that we're only as good as our supply chains. And for oncolytic viruses, this is clearly a primary consideration. But I would also say it's our strong belief these are solvable problems. We don't need to invent new science here. The more formidable challenge is the technical one of optimizing the clinical effect of the agents. I think the manufacturing side is something that is addressable.
Thank you.
I mean, from a manufacturing perspective, for an oncolytic virus, it's really exactly the same as a live attenuated vaccine for an infectious disease.
Yeah.
Isn't, in inverted commas, rocket science, but it is something one needs to know how to do. It is fully scalable. It's entirely off the shelf. And the formulations which are now used provide reasonable stability at 2 to 8 temperatures rather than requiring minus 20 or minus 70 refrigeration.
Well, and importantly, I mean, thankfully, we're not trying to inject billions of people, right? I mean, that the challenge imposed by COVID-19 had so much to do with scale. One would hope to have a broadly useful oncolytic virus that could be used in hundreds of thousands or perhaps a few million patients on a per year basis, but probably not too much more than that. And that means that essentially all of the tools are already in hand to get that done. It's a matter of faithful observance of GMP.
Yeah. And I was wondering if you could answer another question, which is the question of clinical trials. You've been involved in clinical trials, and clearly in skin, you have the ability to obtain longitudinal biopsies to ask questions on whether the oncolytic virus is doing what it's doing. How would you change, or how do you envision clinical trials going on for this disease as well as for the use of oncolytic viruses?
I think it's important to do serial research biopsies of the injected and non-injected tumor tissues. And in cutaneous malignancy, we're fortunate that tissue is often accessible for that type of work. Tumors that involve the skin directly or the subcutaneous can be accessed with punch biopsies, and then a lot of tissue is available in a punch biopsy because the volume is larger than a core needle biopsy. What I think is really interesting, we don't have a full understanding of predictors of response to oncolytic viral therapy. But what I think is interesting is it doesn't seem to be the usual suspects. The tumors that are hot with high tumor infiltrating lymphocytes and high PD-L1 expression, that's not necessarily what's needed to turn on a response and result in a clinically apparent tumor response in a patient. There have been the phase 1b study of TVEC and Pembro, and the phase 1b study of VL-937 and Pembro. The patients who had cold tumors were just as likely to have a response as patients who had hot tumors. So I think the field is still wide open, and I think it's important to continue to do clinical trials in patients for whom serial biopsies are possible. So all different skin cancers, and also things like locally advanced breast cancer.
Thank you. Where will the next advance come from? A question for everybody. And maybe let's start off from Rob. We have about a minute left.
I have to say I'm not myself convinced that these things will ever be used systemically. I don't believe the biological hurdles to achieving systemic administration are necessarily solvable. I do think what we need to be doing is injecting the most potent things we can directly into tumors to kill those tumors as effectively as we can, and then induce the most potent systemic immune response to the released antigens that we can as well. And certainly at Replimune, that's what we've applied to our design of our series of sequentially more potent oncolytic immunotherapies, and particularly taking advantage of the fact that they are also gene therapies with the ability to deliver multiple genes into tumors as they die, aiming at killing the tumor better, providing more immunogenic cell death, particularly adding on things like anti-CTLA-4 or co-stimulatory pathway activation into the tumor to really get each of signal 1, signal 2, and signal 3 in immunogenic terms really going to provide the maximum possible induction of immune response that we can. We clearly don't know if the immune system is actually sufficiently able to kill off truly immunologically cold tumors, but that's what we're going to try to do with our therapies and see how far we can go.
Thank you. We are running out of time, but I would like to thank all our panelists for this interesting session. That's really is a great start for this oncolytic virus session. Thank you.